Hi, everybody, and welcome back to Fossil Friday Web Chats, now hosted on YouTube. I'm Brittany Stoneberg. I'm from the Western Science Center. Joining me, as always, is Gabriel Santos from the ALF Museum.
Hi, everybody.
And today we are joined by Ana Valenzuela Toro. So thank you so much, Ana, for joining us today.
Of course. Thank you for inviting me.
Yeah, I,
We're so
I,
excited.
I am excited to learn about seals, especially elephant seals. They're huge.
Yes, they are.
So, a little bit about Anna. She is a Chilean PhD candidate at UC Santa Cruz, studying the foraging ecology of living and fossil pinnipeds, a group that includes seals, fur seals, and walruses. More marine mammal representation here on this show. She is also a Peter Buck Predoctoral Fellow at the Smithsonian Institution. Other ongoing research projects are in pinniped taphonomy and the paleobiogeography and systematics of fossil pinnipeds from the Northern and Southern Hemisphere. So we're going to learn a lot about elephant seals today.
Woo!
So Anna, whenever you are ready, go ahead and take it away. As a reminder to everybody, if you have questions, go ahead and put them in the Q&A chat, and we will have a Q&A afterwards. So whenever you're ready, Anna.
All right.
All right. Thank you so much, Brittany and Gabe, for inviting me. I'm so honored to be here.
So
I will
today
tell in the
I will tell in the
story of the
story of the
enigmatic case
enigmatic
of a
case of
southern
a
elephant
southern
seal
elephant
from
seal from
Indiana in the
Indiana
Midwest
in
of
the
the
Midwest of the
US.
US.
But before,
But
I will
before
like
I would like to take a few minutes
just
to introduce
myself and my
myself and my
research.
research.
So, as
So, as
Brittany
Brittany
say,
say, I'm
a
a
paleontologist. I'm
paleontologist.
from
I'm from
Chile in
Chile
South
in South
America,
America,
and now I'm
and now
doing
I'm doing
my
my
PhD at
PhD at
UC
UC
Santa
Santa
Cruz here
Cruz
in
here in
California.
California.
For my
For my
research,
research,
I
I
study
study
fossil and
fossil
living
and
marine
living marine
mammals,
mammals,
especially
especially
pinnipeds. I
pinnipeds.
will
I will
explain what
explain what
pinnipeds are
pinnipeds
in a
are in a
few more
few more
minutes.
minutes.
I know
I
this
know this
picture in the
picture in the
background
background
is
is
kind of
kind of
dark,
dark,
but I
but I
really love
really
it.
love it.
This
This
picture was
picture was
taken by
taken by
Gabe
Gabe
a few
a
years
few years
ago
ago
at what is
at what is
called the
called the
Warehouse of
warehouse
the
of the
Natural
Natural
History
History
Museum on Los
Museum on
Angeles
Los Angeles
County.
County.
This is
This
the
is the
second
second
largest
largest
museum
museum
collection of
collection of
marine
marine
mammals in the
mammals in the
country.
country.
And I
And
love
I
this
love this
picture
picture
because of
because
that.
of
It
that. It
combines two
combines
of the
two of the
things I
things I
enjoy the
enjoy the
most,
most,
museum
museum
collections and
collections
marine
and marine
mammals. And
mammals.
I will
And I
tell
will
you
tell
why
you
is
why
that.
is that.
So,
So,
as
as
I
I
say,
say,
my
my
research
research
focuses
focuses
on
on
pinnipeds,
pinnipeds,
and
and
pinnipeds
pinnipeds
are
are
this
this
group
group
of
of
semi-aquatic
semi-aquatic
marine
marine
mammals
mammals
that
that
include
include seals,
seals, fur
fur
seals, and
seals,
sea
and sea
lions,
lions,
and
and
walruses.
walruses.
Pinnipeds
Pinnipeds
evolved from
evolved
a
from a
terrestrial
terrestrial
ancestor
ancestor
likely
likely
30
30
million
million
years
years
ago,
ago,
and
and
since
since
then
then
they
they
have
have
maintained
maintained
this
this
dual
dual
lifestyle.
lifestyle.
That
That
means
means
they
they
forage in
forage
the
in the
water,
water,
but they
but they
breathe
breathe
and
and
nurse
nurse
on
on
land.
land.
And that is different from other fully aquatic marine mammals like whales, dolphins, or sea cows, which spend their whole life at sea. As semi-aquatic marine mammals, pinnipeds have evolved very unique adaptations to deal with this dual lifestyle. Furthermore, modern pinnipeds are top predators. That means that they consume large prey and they play important roles in maintaining the trophic food webs and the marine ecosystem as a whole. So, studying the living and the fossil pinnipeds is essential for addressing broad questions about evolution, but also to explore how diversity, morphology, and even function have changed throughout the evolutionary history of this group. But even more importantly, they allow us to understand how marine ecosystems have changed through time in response to environmental change. So for that reason, my research combined both fossil and living pinnipeds to investigate the evolutionary history of this group, but also how their evolution is linked to changes in the marine ecosystem over time. And to do that, using different approaches, including comparative morphology and ecogeochemical analysis of living and fossil pinnipeds. So in this picture, I show one example how comparative morphology of fossils has helped us to understand better how the diversity of marine mammals have changed over time. So based on morphological comparisons, we were able to identify and describe a new species of a tiny seal that lived around 5 million years ago along the coastal margins of Chile and Peru in South America. And why this is interesting? Well, this is an area where seals are absent today. So that demonstrates that the occurrence of very important changes in the marine ecosystem during at least the last during the last 5 million years. So, by the way, this is a humerus, an arm bone of this tiny seal, and those are my hands. So, well, here is another example, which is part of my dissertation research that I'm performing right now. This is still work in progress, and here in this project, I investigated the foraging ecology of coexistent species, sympatric species of fur seals and sea lions from the Northern and Southern Hemisphere. And basically, like here, I'm trying to answer the question of how these animals that live together are able to coexist. So for understanding this, I'm performing ecogeochemical analysis of modern bonds. Basically, I'm performing stable isotopes on them. And this type of studies is relevant not only because we can get a better understanding of how modern marine mammals communities function or work, but also help us to make more informed inferences or about how fossil communities of marine mammals functioned in the past. So, but what do all these projects have in common? So they are all based in specimens from museum collections. Museum collections are important because they archive specimens which provide snapshots of species or communities at a particular time or space. And that information will be lost if we didn't exist in these collections. So this picture here is one of the largest collection. Here in this picture, I'm showing one of the largest collections of marine mammals in the world. And this is located at the Smithsonian Museum Support Center in Maryland, in the East Coast of the US. And here you can see there are 1,000 specimens, including the ones of a blue whale. And here you can see the column vertebra of a blue whale too. I'm here and there as a reference. So pretty cool. This is an awesome place to be. Okay. So here is another example of a spectacular museum collection. These are skulls of California sea lions at the California Academy of Sciences in San Francisco. This is, to my knowledge, the most extensive ocelogical collection for a single marine mammal species in the world. Each of those specimens has a unique story related to the particular time and place where it lived, right? So therefore, each museum specimen has a story to tell, and it's our job as a researcher or as a paleontologist to unravel them. And today, I will present one extraordinary story that came out from an unexpected museum specimen. So now we are gonna talk about elephant seals. So elephant seals are the largest living pinnipeds, and males can reach 5 meters long or 60 feet if you want. Males are also characterized by their large proboscis or nose, which can be used to produce very loud sounds, especially during the breeding season, when mating competition, as you can see here, occurs. Elephant seals are also excellent divers, spending almost 90% of their life at sea. So today there are two species of elephant seals, northern elephant seals and southern elephant seals. Externally, they look very similar. Northern elephant seals are distributed around the North Pacific, mostly, you can see here. In contrast, southern elephant seals are distributed in the Southern Ocean and some adjacent islands. So you can see here Antarctica, so southern elephant seals are distributed around the Southern Ocean. But before going any further with this story, I would like to acknowledge the co-author of this work, Maria Sikos and Nick Pinson. Maria is a PhD student at Queen Mary University of London, and she's studying the evolutionary history of sloths. And Nick is the curator of fossil marine mammals at the National Museum of Natural History at the Smithsonian in Washington, D.C. They are the true heroes of this story, and nothing will be possible without their hard work and perseverance. So, having in mind about what I say, that elephant seals are marine animals that live along the North Pacific or in the Southern Ocean, you will never expect to find an elephant seal from the middle of the continent, right? But that is precisely what happened. So hidden in the shelf of the museum collection at the Smithsonian, they were discussed. This is a replica of a skull of an animal that appeared to be an elephant seal. We say that we infer that it was similar to an elephant seal, mostly because of the size of this canine. So to give you a references, this is the part of the rostrum, like this part of the skull, and this could be the canine. So, okay, but there was something wrong about the specimen because all the documents associated to this specimen say that the original specimen came from Indiana in the Midwest of the United States. So something was odd in this case. So here you can see a better view of this specimen. So this is the anterior section of the rostrum, and this is going to be the canine. And this is a ventral view. So basically, you're looking below, and these little holes are basically the alveoli, is where are the pockets where the teeth are inserted. So this is a pretty cool specimen. It's fragmentary, but it's a very cool specimen. So the documents that were associated with this specimen also reveal there was an additional cast of this same specimen at the Field Museum in Chicago. The archival notes associated to this second specimen in Chicago included some original correspondence between important museum curators and researchers from the 70s, which were all intrigued by this mysterious specimen. So remember, the 70s, they didn't have email or internet, so all these communication were by mail, like physical mail. So these documents also reveal something very cool, is that this original specimen was accidentally found and collected during a building excavation in 1965, and it was found approximately 9 meters below the surface of the riverbanks of the Wabash River near Lafayette in Indiana. But very soon we realized that unfortunately the original specimen was destroyed for carbon dating analysis in the 70s. And these tiny chunks I'm showing here are the only remains left from the original specimen. And the radiocarbon dating resulted in an estimated age of 1,200 years before the present, time before the Europeans arrived in the Americas. So we were super intrigued by this specimen, and because we only had a cast of a part of the skull, we relied solely on morphology to identify this specimen. So we went back to the collections and started taking out elephant seal skulls, but also skulls from other pinniped species from cabinets. And by comparing the cast with this other skull, we were able to determine that this mysterious specimen actually represented an adult male elephant seal. So, but we wanted to go farther, and we started to compare in more detail this cast with the morphology exhibited by Northern and Southern elephant seals. And despite the external similarity of Northern and Southern elephant seals, they slightly differ in body size, but also in skull morphology. Southern elephant seals are somewhat larger and more robust, and they have a more robust than the skull of Northern elephant seals. Those differences are reflected in the relative position and elongation of different structures in the anterior section of the rostrum. Therefore, by comparing the cast with many skulls of Northern and Southern elephant seals from different museum collections, including California Academy of Sciences, the Smithsonian Institution, and others, we concluded that this mysterious specimen, actually, this specimen from Indiana, actually corresponded to adult male Southern elephant seal, like the one I'm showing in this picture. So, very cool. Furthermore, when describing and comparing the specimen, we notice that it has some marks that I'm showing here, these arrowheads, that look like intentional cuts. This potential cut mark suggests that the original carcass might have been butchered by indigenous people before it was buried. However, the archaeological record also tells us that only skeletal elements of high socioeconomic value, like isolated teeth or canines, will likely be transported long distances and traded by indigenous people. Therefore, it seems unlikely that a complete skull was traded from South America to North America. Also, the archival notes that I mentioned before noted that the original specimen had some black pebbles and sand in one of the alveolus of the teeth, or this teeth pocket that I showed before, which are those are indicative of some time of exposure to water currents, suggesting that the specimen underwent some type of post-mortem river transportation. Furthermore, the complete lack of carnivore scavenging marks suggests a relatively short post-mortem period before its burial. So it was very quickly buried after the animal died. So now the big question: how did this male southern elephant seal arrive in Indiana, which is more than 10,000 kilometers outside its known distribution range in the Southern Ocean? Is that even possible? So to answer those questions, we have to look back in more detail the biology and life history of the living elephant seals. So elephant seals are a remarkable species because these animals are excellent divers and they can perform very long foraging migrations every year, which can extend several thousand of kilometers, spending on average almost 10 months of the year at sea. But how do we know this? Well, they are marine mammals, so we don't know much about their biology because they left the coast and we don't see them anymore. So we know this mostly because of the fantastic work performed by many scientists and engineers who have developed a wide range of sensors, as the one I'm showing here in this northern elephant seal. You can see here this in the head and here this in the back. The sensors collect data about the ocean, but also about the animal's behavior. So these sensors are designed to fit in an instrument about the size of a credit card. They are very tiny. And these instruments can be attached to the head of the animal or in other parts of the body, allowing to researchers to gather information about the animal's behavior. So imagine this as having a GPS in your head, and it's tracking you. So you might wonder whether this instrument hurt the animals. Well, the answer is no. So these instruments are attached on top of the seal's head over the fur, and northern elephant seal or elephant seals in general molt. That means that they lost their fur every year. So the device will eventually fall off next year when the animal shed its coat. So now that we know that elephant seals are excellent divers, excellent ocean navigators, and spend most of the time at sea, and then we start looking in the literature and we realize that over the years, they have been reported several extraliminal occurrences at very distant locations from their known range. So here in this map, I'm showing you the distribution of northern in purple and southern in orange elephant seals. The big dots represent the principal breeding colonies of elephant seals, here in the northern elephant seals and southern elephant seals. And the small dots represent these extraliminal occurrences of southern elephant seals along the Pacific, but also along the Atlantic. So here we can see that background southern elephant seals have been found in many places, including in Argentina, Uruguay, Brazil, Chile, Peru, Ecuador, and even Panama. But I want you to bring your attention to this very small dot here, that almost invisible. This corresponds to a southern elephant seal that arrived alive and healthy early this year to the Gulf of California. And here I show you this animal. So this is a male southern elephant seal that was reported early this year, in 2020, in the Gulf of California. And this is a close-up of the hind limbs. And you can see, if you look with attention, you will see that these little things are barnacles. So for that to happen, that means to have barnacles in the hand flipper, this animal should have spent a lot of time in water. So yeah, this animal crossed the whole Pacific, crossed the tropics, and then got into the Gulf of California. It's a very impressive animal. Furthermore, we don't have to forget that this southern elephant seal was found in the Wabash River around 1,000 kilometers from the closest marine source, and potentially represent the most extreme freshwater incursion of a marine pinniped ever recorded. Notably, inland Holocene marine mammals have been previously reported across the US, including manatees from Ohio and the Mississippi rivers, as well bowhead rorqual whales and sperm whales from Michigan. Also, there is a single record of a vagrant elephant seal, southern elephant seal, from the Guayas River, here in the Gulf of Guayaquil in Ecuador. And this case suggests that there is no apparent physical barrier for marine mammals to invade and make long-distance upriver incursions. So this is super, super cool, in my opinion. So based on all these records, we suggest, considering the biology of elephant seal, we suggest that this male southern elephant seal crosses the equator and basically entered the Gulf of Mexico and then entered into the Mississippi River system stranding far upriver in Indiana, where some indigenous people might found the animal and potentially kill it, or maybe found it dead. And they butcher it and transport it for a short distance to then discard the remains into the river. So, but why this crazy story is important? So this occurrence shows us that the dispersal behavior of elephant seal is more dynamic and more complex than previously recognized, suggests us that reconsideration of the current assumptions of elephant seal life history patterns, and it stresses the need to think more broadly about the ecology and evolution of this group. Furthermore, both southern and northern elephant seals suffered from extreme hunting in the 18th century, and today they are facing another obstacle, the climate change. And by learning about this species range behavior in the past, predict the many ways that elephant seals could react to the current global climate change. So, and with that, I would like to thank all these institutions for their support, and thank you all of you for your attention. And if you want to read more about this crazy occurrence, please check out the paper. It's open access, so you can download it for free. So thank you, everyone.
That was fantastic.
No.
The Robo Seal coming up.
Yeah, by the
Robo
way, that
sees all.
Yeah, by the way, that is the main research topic in my one of my lab here in UC Santa Cruz, the Costa Lab. They perform this amazing research where they put tracking devices on seals, on northern elephant seals, and they have been able to recognize this broad distribution range of northern elephant seals and these very long migrations that occur every single year over time. So it's super amazing.
That's so cool. There's so many things we can learn, right? From like tracking like marine mammals through their life and like following them in the ocean is where all that stuff, right?
Yeah, there are many things. I mean, marine mammals, we have been studying marine mammals for a long time, but we still are learning about them because they spend most of the time at sea, right? And we are here. So yeah, by designing these new techniques, new methodologies, and also combining with fossils, we are learning a lot about them, about how they evolve, how is their ecology, and etc. So it's super amazing.
For you, why do you think ocean-going marine mammals will swim up like rivers and things like that? What do you think the cause is?
That is an excellent question. So, some people believe that these extraliminal occurrences outside the known range is related with changes in the prey distribution. So, because of this El Nino, La Nina events where the water changes the temperature and that affects the prey distribution. So that basically what happens is the marine mammals eventually will follow the prey and will, because they are following, they will, I don't know, go to places where they don't usually go.
Interesting. I guess,
Yeah, but it's still I mean, there's a lot of research trying to figure it out what is happening.
yeah, I remember seeing on the news there's always like a whale or like something that goes upriver, and they're always like, how is this here? It's such a weird thing to see. I know for most people too.
No, yeah, indeed, it's super weird. I was reading an article that came out, I think, last year in newspapers about harbor seals that were breathing in the Thames in London, like in the river, and they were breathing there. So it's crazy.
Yeah, here in Los Angeles, we've get I think green sea turtles that swim up some of our flood channels because like the water is warmer, and so like if you're like taking like running down the trail or the bike trails, if you look, you can see green sea turtles like all the way
Wow.
inland from the ocean, and it's really cool. I have not seen one myself, but that's because I don't really exercise. But eventually, I'll get out there and see them.
All right, here's a question from the Q&A. This is from Derek, who's asking, can you elaborate more on the records of cetaceans in Michigan? Says it sounds wild.
Oh yeah, I know, it's super wild. So when we were working on this in this project, we started looking at the literature and we found this very anecdotic and like small mentions in different papers that mentioned that, oh, of course there was a whale in Michigan. So there were these are like osteological remains that were found in different places in different areas, but there are not much research about them. I know that some of those specimens are in museum collections, so we need more people to look at those specimens and maybe perform some, I don't know, a stable isotope analysis to confirm that the occurrence and actually understand what those animals were doing there. But of course, we cannot discard that there were some type of human transportation because these specimens are kind of old, so probably there was some kind of transportation mediated by humans at some point. But yeah, we don't know much about it, unfortunately.
I know some people might be wondering, because here in California and in a lot of other places, we find marine mammal fossils in like mountains or like really far inland. Is that the same reason why we're finding those fossils there, that they swam upriver, or is it for a different reason?
Yeah, so it will depend, but I think if most of the fossils that we can find in mountains that are marine, it has to be because the geological configuration of land have changed over time. So, for example, if you go to South America and you see the Los Andes mountain, this very long mountain, is full of ammonites or other marine invertebrates. And it's not that means that in the past, these mountains were actually under the water and all the geological configuration of the land have changed because of, I don't know, many things that happen over time. So yeah.
For us marine mammal people, that's really lucky that we can find the fossils out here.
Yes, I know.
How did you get started in studying marine mammals?
Oh, so I would say that I was doing my undergrad and I took a class about comparative anatomy, and we went to a museum, the Natural History Museum in Santiago, in Chile. And then I met one marine mammal paleontologist, Carolina Simon Gottstein, and we were talking. You know her. We were talking, and she showed me the collection, and I was oh my goodness, I think that changed my life. And I seeing those fossil marine mammals there, and she told me the story how they occur in that area. So I was completely crazy, and since then I started thinking and studying about pinnipeds, marine mammals in general. Yeah.
And that's how we met too, right? Because you were coming to visit the Natural History Museum, and that's when I took the picture, right?
Yeah, I know. That occurred in 2014, I believe. That was my uh, first time in the US. And we went to this collection in Los Angeles. And yeah, and you helping me with taking pictures and measurements and everything there. It was super fun.
Yeah.
I love that trip. Yeah.
Oh, we had a great time, and seeing that collection was
Yeah.
awesome.
Oh,
Yeah,
goodness.
here's another question. This is again from Derek. Why did the entire maxilla have to be destroyed for carbon dating?
Oh, yeah, so this specimen was sampled in the 70s. So during that time, the technology available for doing this kind of sampling wasn't developed enough. So we needed a lot of material to just get the sample analyzed. So that is why they probably decided to put all the material there and yeah, and run the analysis. But today that would be crazy. I mean, today we only need like a tiny, tiny, tiny sample of the specimen to do that. But hopefully, yeah, the animal people in that time was super intelligent and smart, and they created this cast. And so now we are able to reconstruct this crazy story.
That's why it's really important for museum collections to make replicas and digitize and, have the fossils, because sometimes we have to not destroy, we consume a part of it. As my curator, Dr. Andy Farkey, says, we do consumptive sampling. So it's always important to have backups.
No, of course, and not only because we can consume the specimens, but also accidents can happen. So think about the fire that occurred in the Natural History Museum in Brazil a couple of years ago.
Yeah.
I don't know, we are here in California, and here we can have an earthquake at any time or a fire. So having replicas or casts in other institutions is super important because we are able to preserve the information over next generations. And yeah, and I think more effort should be put in preserving collections over time.
Absolutely. For in our museum, as a collections manager, like, we're always trying to digitize. I was just actually scanning a turtle skull this week for a student to study, and now we have this complete digital skull ready to send to anybody who needs to study it within like a moment's notice, and they don't even have to come to our museum.
Yeah, the Western Science Center does the same. We have a molding and casting lab. We have a 3D printing lab. Whenever we publish, we try to put those scans online for people to see. So yeah, it's a very important component of museum work.
Yeah, absolutely.
Here's a question. Did seals start out as freshwater mammals and evolve to return to the ocean?
Okay, yeah, this is a hard one. So we usually thought that, traditionally we thought that pinnipeds originated in marine environments because the oldest specimen came from marine sediments. But time ago, it was published a new specimen, Pygila darwini, that came from freshwater sediments in Canada. So we are now debating whether pinnipeds originally originated in freshwater ecosystems and then migrated into marine, or if they originated in marine ecosystems already. So, but it's an excellent question.
The transition from land to water is like one of those big questions. We had Kirsten Formosa on the show previously when we were on Facebook, who talked about secondary adaptations from land to water, and that was a really cool episode if you want to check that out.
Yeah, I will, definitely. Yeah. This transition is super interesting and different. And what is more interesting, that different taxa found different solutions to that transition. And that is why pinnipeds, they are in the limbo. They are not fully terrestrial, are so important to answer these questions.
Yeah, for me, since I studied desmostilians in the past, desmostilians were marine mammals, but they still looked very terrestrial. They didn't evolve flippers or anything, but they still were out there in the oceans.
Yeah. Absolutely.
Oh.
Yeah, I'm thinking when
Yeah.
you told me about this, I always think about Thalassos because the Smithsonians are this group that live here in California in the North Pacific. But in the Southern Hemisphere, we didn't have the Smithsonians, and we have an aquatic sloth that it was looked like a sloth, a huge sloth, but it was marine. So it's super cool to see this contrast and how different animals and different taxa have found their solution and they enter into the water.
Yeah. I got to see a skeleton of Thalassos when I was in Peru, and I just wanted to stare at it for like hours because it's so weird and so cool.
I know. Yeah.
Oh, and I did want to say all those questions, which were really good questions, came from Derek, who says they have a group watching and they had lots of cool questions. So thank you, Derek, and thank you, your group for all those excellent questions. So, let's see. And somebody says they really wish they could study Thessalon. Oh, I'm gonna mispronounce it.
Thalassos.
That one, yeah.
Yes.
Well, awesome. So, are there any other questions on your end, Gabe?
No, I don't think so. Actually, let's so for that comment that Maria put, they want to study Thalassochnos for ancient DNA. Anita, do you want to comment on why there might be hard to find ancient DNA in Thalassochnos?
Yeah. So DNA is a very sensitive molecule, and basically DNA disintegrate over time. So in this molecule, Thalassochnos are very old. We are talking about animals from, I would say, 5 million years ago, even older in some cases. So those fossils do not preserve DNA. So that is why it's so hard to make this analysis more complex. But that is why comparative morphology is so important, because just observing the bones or the skulls and different things, we can reconstruct and understand the life history and the evolutionary history too. Yeah, super cool.
Yeah. Awesome. Well, thank you everybody for your questions, and thank you, Anna, for all of this really cool information and for presenting it to us today.
No, thank you so much to you, a lot of you, for inviting me.
Of course. We'll be back next week, as always, at 10 a.m. And Gabe, did you have any closing remarks?
Yeah, like Brittany said, join us next week. Next week we have Dr. Ali Nabavizadeh, who'll be talking about extreme dinosaur heads. We're going to be looking at like some of the really cool adaptations that herbivores had in dinosaurs. So stay tuned for that one. If you want to keep up with all of our Fossil Friday Chat episodes, make sure you subscribe to our channel and turn on notifications for every time we upload a new episode. And we also want to thank webcaptioner.com for providing the web captions for the show today. I just want to say thank you again to everybody for tuning in, and thank you, Anita, too.
Yeah, and as always, if you'd like to support either of our museums, the Western Science Center or the Alf Museum of Paleontology, this is a joint program between the both of us. If you'd like to support us, there will be information on how to do that, and you can go to either of our websites.
Oh, yeah, and that information is in those are the links in the description below, and we'll include Anita's talk or Anna's talk in there as well, or her paper. Sorry.
It's okay.
It's Friday.
It's Friday. I'm ready to go eat lunch or something.
Yeah. Thank you, everybody, and we will see you next week.